Okay, so following up on Bob's talk regarding using whole genome sequencing to call copy number variation, I'm going to discuss today how we use whole exome sequencing, and he mentioned a little bit of the difficulties, some of the difficulties involved in using whole exome sequencing for calling copy number variation. So for those of you that don't know me, I'm Menachem Fromer. I'm now actually at Icahn Medical School at Mount Sinai. I started my genomics career here at the BroadE, and I'm still part of the Stanley Center here for psychiatric research. And I'll mention some of the data that we've used in sort of prototyping and running the copy number variation tools that we've developed on psychiatric disease data. So our tool is called XHMM, and that's for exome hidden Markov model, and I'll go into details about that now. So just, I don't think I need to go through this there in a lot of detail after Bob's talk, but just in terms of the way we view it, because I'll have a sort of different cartoon later on. When we have copy number variation, so in the reference you'll have, again, we're dealing with exome sequencing, so our focus is on genes. So just in this cartoon, you have in a reference genome, someone that's diploid, you'll have 2 copies of the gene. In a deletion, you'll have maybe 1 copy of a gene. And a duplication, in this case, a tandem duplication, you'll have 2 copies of the gene on one of the chromosomes and 1 copy on the other. You'll end up having 3 copies of the gene. Exome sequencing, for those of you that are familiar or not familiar, just a quick rundown. The point of exome sequencing is to literally, instead of sequencing the whole genome, you're sequencing the functional parts of the genome, or at least the protein coding parts of the genome, or the exome, because you're really sequencing the exons. And the way you do that is using this array capture. So it's a hybridization array, where you capture, using reverse complementarity, you capture the protein coding sequence, and then you enrich your DNA fraction, your genomic DNA, to be mostly exomic DNA, and then that's what you're going to be sequencing. But as Bob mentioned before, one of the main issues is that besides all of the biases inherent in sequencing, in terms of what you're going to actually see, what you're going to be able to map, this extra capture step adds extra biases in the read data that you're going to be seeing, the sequencing data. So we have to deal with that in the exome sequencing, as well as some other issues that I'll mention shortly. So our goal was to use the exome sequencing to accurately call copy number at exon level resolution. So as opposed to genome strip, where in genome strip Bob was describing how you actually call it base pair resolution, at least for now, XHMM, what it's focusing on is using exome sequencing to call at exon level resolution. So for each exon and each individual, you'll be saying, is that exon deleted, duplicated, or diploid? So you wanted to talk about those levels and not at the base pair resolution. Again, not yet. We do have some plans that we're working on, but that's very preliminary for now. So I'm going to talk about what we do have. And the main sort of metric, the main feature of the exome sequencing that we use is the read depth. And the reason for that is, in terms of the other possibilities, in terms of using the read pair information or split reads, the information, as Bob mentioned, is going to be very sparse for exome sequencing, right? So the exome is basically the union of all exons, and maybe some regions nearby, but the exons are very clustered together in the genome, but then there's large intergenic gaps in the genome that aren't going to be captured by your exome sequencing. So you have lots of small regions, relatively small regions that you are sequencing, and they're nearby each other, and they're clustered, and then there's a big gap, and then you have that again. So in terms of, first of all, it's very sparse data, so the chance of actually having any particular breakpoint in the exome is kind of low. So at least for now, we've prioritized using read depth information, which you do have throughout the exome, as opposed to the split read sort of breakpoint focused approaches. And the same thing for the read pair also. The read pair, you're not going to have read pairs that necessarily map far away or close by in the exome, because you're only trying to sequence the exome itself. So again, our focus is going to be on using read depth to extract from exome sequencing data the copy number variation that exists. So just a general run through of the CNV calling pipeline that we actually developed. And I should say that the talk today is broken into sort of two parts. One where I'm going to give sort of a higher level overview of the methods, and then the second part I'll go through actual practical examples of command lines and how you actually run them. Not in too much detail, but just to give you some feel of how you actually use the XHMM tool set. So in the CNV calling pipeline of XHMM that we developed, the first step, and this is the most computationally intensive step, is actually just calculating the read depth for each exon and for each sample in your data set. And we do that using GATK. I'll mention a little bit later about a Q script that we have that runs this whole pipeline, but there's also steps in the tutorial, and I'll go through each step where you run GATK, and then you run a bunch of different XHMM modules to actually get through this pipeline. Then I'm going to focus now on some of the main steps. Those are the ones highlighted in blue on the slides, but for reference, I put the other ones in, and they also correspond to later what might be in the slides. The next main step is, because of the different biases introduced during sequencing and before that during exome capture, in terms of what read depth levels you'll expect for different targets and for different samples. So we actually need to normalize the read depths before we actually want to use those read depths to be indicative of copy number. And the way we normalize the read depths is using principal component analysis or singular value decomposition. And what we actually do is remove the top principal components. Again, each of these steps I'm going to go through in some detail later. And then the next step in blue, which is the next main step, is once we've normalized the read depth data, we actually want to use a hidden Markov model to call CNVs across exome targets, or across exons, basically. And we're effectively merging the targets into across the introns, we're calling the CNVs in the exome. So just in terms of calculating depth of coverage, so this is just an IGV screenshot for those of you that aren't familiar of showing what the reads look like in exome sequencing. And literally what we do here is pretty simple, but I'm just going through it to be clear. Literally what we do, because there are different strategies for read counting. And what we actually do is we'll look at an exon region, meaning an exon target, and we'll actually count for each of the bases in that exon. We'll count the number of reads covering that base, again, with some filtering on base quality and mapping quality. And again, we'll talk about that in some of the details later on. But we'll count how many bases map to each position in the exon, and then average that. And then you get one number for that exon for that particular sample based on that sample's reads. In the end, if you actually just visualize what you get from running depth of coverage, what you get is some sort of matrix like this, right? So you have on the rows here are individuals, or the samples, and in the columns here you have different exome targets. In this case, almost 200,000 exome targets in a sort of typical capture here at the BroadE. And what you see is that there are lots of patterns already, right? So you see that, again, their columns are the targets, so you'll see that there's some targets that drop out that have white, that are pretty white, and those are basically correlate to targets that have very low depth. So already you see that there's some patterns like that. There are, if you look all the way at the very right, there are the sex chromosomes. Here it's just the targets are sorted by chromosome. So you see that there's sort of like a different pattern there, and that's based on sample-specific, right, gender. Males have only one copy of chromosome X for the non-pseudo-autosomal region. So that's the banding you sort of see all the way to the right. And you also see certain, if you look at the rows, there are certain patterns there. You see that there are certain samples that were sequenced at higher coverage, and those samples are going to have higher depth. Of course, having higher depth, as you see here, doesn't necessarily mean you have a CNV, or lower depth doesn't mean you have a CNV. We need to sort of normalize away these different effects, the ones we know about and possibly ones that we're not as familiar with, but we would want the data to sort of drive that normalization. Just as an example, a zoomed in sort of example of what this type of data actually looks like. So here what we have on the x-axis is genomic coordinates for almost 200 kb. It's 26 targets, 26 exons. And what you see, there's a bunch of different genes, but here it doesn't even matter which genes necessarily they're coming from. And what you see on the y-axis is just unnormalized read depth, literally the output from GATK depth of coverage walker. So what you actually see, and each gray line here is a different sample. I think there are approximately, I don't remember the exact number, maybe a few hundred samples here. So what you actually see is that each gray line is a different sample, and there's lots of variability. Some samples seem to have higher coverage than others, but even a stronger signal, I would say, is that between these different exome targets, which are those black dots correspond to the exome, the 26 exome targets, you see that there's very large variability. So some targets just have higher coverage, they have higher capture efficiency, they have higher sequencing efficiency. Either way, you're seeing a lot more reads at those targets, but it doesn't mean that those targets are really different in copy between other exome targets, between other exons. So you need to normalize that out, and here we've colored in green some samples and their corresponding targets that we think might be duplicated. Question is, what do we really think if we try to normalize the data, clean it up a little bit for these sorts of batch effects? So what we'd like to do, and this is just a real example from the algorithm that we actually use, is clean up the data so that most of the samples presumably are going to be copy number 2, and they should be pretty flat in terms of whatever scale you're looking at. They'll be pretty flat and pretty straight, and then the samples that have duplications in those regions, you'll actually see in this case, they'll be above the line, indicating that they have higher than average copy number, or read depth that corresponds to having increased copy number, or in this case, duplication. Right? And the way we actually do that, we normalize data, is using principal component analysis, PCA, and just conceptually what it does is rotates the high dimensional data, that matrix that we were looking at, and tries to find some underlying structure. And the structure doesn't have to be known, but it does have to be linear, because that's the way at least principal component analysis works. There are some similar tools, including Conifer, and other tools that do some similar normalization. It seems to be a pretty good choice of methods. We basically implemented this algorithm starting in 2011, and some other groups did also. It seems to be like the method of choice in exome sequencing, actually, for normalizing data. But there are other methods, but I'm not going to talk about those today. The important point is that what you gain from using principal component analysis is that what we think basically is that, at least for exome sequencing, it's kind of difficult to exactly come up with a sort of generative model that tells you what to expect for different GC by GC content, for example. But you can see that the data actually has this GC bias. So what you actually see here, for now, this isn't actually exome capture yet. This is just saying for the exons in the exome capture, so you have 200,000 points here across the genome. These are genomic coordinates, so it's every single exon here. You actually just see on a scale of 0 to 1, what is the GC content for that exon. But when you do principal component analysis, what we actually see in this particular scenario, and this usually comes out as one of the strongest principal components, you find some principal component that sort of mirrors this the GC content of the exons. So in this case, the third principal component has a Pearson correlation of 0.65. So you're already saying, okay, well, it is capturing the GC bias that we knew would induce certain biases in the exome capture, meaning if you have higher GC, you might have higher capture, and lower GC, you might have lower capture, and that's what it's sort of encoding here in the principal component. But this is just one of the principal components. So what we actually did is we look at all the principal components and we try to understand where are the principal components that correspond primarily to non-copy number factors in the read depth data. And those are the principal components that we actually want to remove from the data. Just another example before I go on to talking about all the principal components together is if you do have both male and female samples in your entire sort of cohort, what you'll actually see, in this case, it was a pretty close to 50-50 split, so it's a pretty strong principal component that comes out. What you actually see is a female pattern of read depth variation. You see that autosome-wide, which is the first 22 chromosomes, right? So you don't actually see any strong signal here in this principal component, but what you do see is that in the X chromosome, and that's the black region at the end, all the way at the right, you see very high read depth, and in the gray chromosome, all the way at the end of the right, you see really low Y chromosome read depth. And what that basically tells you is that this is the female pattern of read depth variation. Of course, males will have the opposite. So this really loads onto female-male differences very strongly. So we see that just doing principal component analysis comes up with sort of these sample batch effects and target batch effects that we expect in terms of GC content and gender, but we also see that it's going to come up with some other, effects, and we want to remove all of those. So what did we actually do, at least in prototyping this? What we actually did was we tried to think of different target and sample genome or exome-wide, really, effects that might be correlated with read depth, including GC content, the size of the target. Is that a known copy number variation region based on the DGV, database of genomic variants, that Bob mentioned earlier? Is there a segmental duplication region? Is there a repeat region? In terms of just the sample that we were dealing with at the time, does it correlate with phenotyping, in this case schizophrenia, or cases or controls? What does gender look like? What are the batches? There were 1,000 samples in this that were actually sequenced at different times in different sequencing batches. Does that correlate? The mean read depth for the sample, mean read depth for the target. The thing all the way on the right is population components. Basically, we threw a lot of different things here to try to get a feel. What does the read depth actually, or the principal components that are the strongest variance in the read depth, what does that actually correlate with? And what we actually see is, if you look on sort of the left side of the plot, which is to the left of the dotted line, there's the strongest principal components because they're sorted by the variance that they contribute to the data, or that the data is basically in that component. And what you see, the strongest correlations are basically with actually the sequencing batch. So certain batches might have had higher coverage, and that's an important factor that you should take into account. What else was important was the GC content, as we saw. Gender was important. But most importantly, but besides batch actually, was just the mean read depth per sample. So even within batch, there are certain samples that have higher or lower mean read depth, and that's a strong component that needs to be corrected for. And we're going to do that using the principal component analysis. So you basically do this principal component analysis, and as the plot says, you actually just remove those strongest principal components. So in this case, you remove the first 18 principal components, and you're going to reconstruct the read depth signal without that strong variance that's due to non-copy number signal. Just looking at this sort of yes, so that's this slide. Good question. Thank you for leading into the question. So yeah, so if you actually look at the components and you plot them in terms of their variance, our hypothesis was, and this seems to hold true in lots of different data sets that we have looked at, including schizophrenia data, autism data, but also other non-disease data also. Basically is that there's a relatively small number of high variance principal components that, and this is pretty typical actually, often of principal component analysis, but especially in this read depth data, that there's a small number of principal components that contribute most of the variance to the data, and after that there's a pretty strong drop-off, and this is a log scale, so really you're seeing that there's a pretty big drop-off here. So, and the y-axis here, the x-axis here is just the principal component, index sort of, sorted by the y-axis values. So you see that in this case, again, around 20 components, there's a pretty big drop-off, and our assumption is that those first principal components, even if we don't know if they correlate directly with things like GC, they seem to look similar enough to those types of components in terms of the way they scatter sort of exome-wide, but also in terms of their variance. So we're assuming that those are not related to copy number, true copy number signal, and more experimental artifacts, batch artifacts, real things about the genome, including GC content, but things that we're not interested when we're looking for copy number variation. And in XHMM, I'll mention that a little bit later, we sort of have a pretty simple rule of thumb that we use to actually get to remove these small number of principal components to normalize the data. So basically, we'll remove this small number of strongest principal components, reconstruct the read depth data, and that will be the read depth data that we call the normalized read depth data that we go forward with and use to actually call copy number variation. So now that we have that normalized read depth data, what we actually do, and this is why XHMM is called XHMM, because we use an HMM, a hidden Markov model, to basically connect the dots between neighboring exons to call the CNVs. Because for any one target, the data is still pretty noisy. So knowing, just saying I have high redepth or low redepth at that target is still a little bit hard to say, does that correspond to a duplication or deletion? But when you start stringing together the exons, and people have done this before for array-based CNV calling, GWAS-based CNV calling in tools like BroadE Suite. And the point is basically, if I see a string, I see a run of exon targets where there is depleted redepth or increased redepth, basically that should correspond either to a deletion or duplication. And this is, again, using the normalized redepth. And one of the things that I should say about the hidden Markov model here is that it takes into account some sort of genome-wide CNV rate, how much you expect, the length of CNVs. Again, these are mean parameters for the distributions, but also importantly, the distance between the exome targets. Again, because we know that exome targets are not very uniform, because for a single gene, they'll be very close together, but then there'll be a big gap from one gene off into the next gene, right? So we want to take that into account in terms of some of the parameters that go into the hidden Markov model. And there's some other related methods. I'm not going to go into that now. So what does it actually look like when you run the data? So you have some normalized redepth data, and that's what's plotted here. Again, genomic coordinates, or actually exons on the x-axis, and the y-axis, you see this is normalized redepth data. And what you actually see, and these are z-scores, which we actually use in practice, but what you actually see is, so if you have a deletion, what you'll have are lots of consistent dots clustered very tightly. In this case, just because we're zoomed out, you'll see that there's a deletion in that region, and we've marked it as red. And there's some duplications all the way in the X, or maybe chromosome 22. And in the Y chromosome, those are regions of runs of very high redepth, normalized redepth. So those are what we want to cause duplications using the hidden Markov model. So, I mean, this is just sort of sanity check what happens when we actually look at the mean redepth. This is actually the unnormalized redepth. When we look at duplications versus deletions, this is sort of just to say that it's not like when we do the normalization, we sort of reverse all the data or do something crazy. On average, the duplications, what the duplication calls, if you look at their original redepth, do have much higher, they do have significantly higher redepth than the deletions. One of the important points is, so we have a hidden Markov model. We'll go back to this slide. We have a hidden Markov model here that calls copy number variants across the exome. Fine, so you have some call. You can mark it in red, mark it in green. Those are just indicating the calls. But in terms of actually knowing how much should I believe that call? Yes? Oh, yeah. This one. This one. Sorry, what did you say? So the x-axis is just the genomic coordinates, right? And the y-axis is this is the normalized coverage in XHMM. So we use the PCA to normalize the coverage for this one sample.
Yes, and this one sample compared with which kind of normalized. So you normalize, you have one sample.
Oh, okay. You're saying how many samples are actually in the batch that we used? So in this case, it was also a few hundred samples. I can't remember exactly for this particular plot.
Like you have average based on like 100 samples, then you use one sample compared with that average of 100 samples, and then you make, I see the middle is 0, so it's not ratio, right?
Well, no, so it's actually a z-score. It's a z-score that's unique for this sample, but the way that we integrated the 100 samples, let's say, is when we did this principal component analysis, it was, going back to, sorry. So we had this matrix of let's say 100 individuals in the rows and 200,000 targets. So at that point, we considered all the samples simultaneously for the normalization. We run PCA on this matrix. And at that point, if one sample would have double the redepth, that would come out just as saying that sample has double the mean redepth. And that would get normalized away. You understand what I'm saying? And that's why after the principal component analysis derived normalization, then that sample wouldn't have that factor of 2 anymore. And then you're able, then we actually call a single sample. Yes, it's a good point. Well, it's also, I mean, we are looking at this pretty zoomed out, right? This is the whole exome. If you zoom into any region, right, there's, do I have the mouse here? Let's see, yeah. I mean, if you look at, there's some points here that are, fine, these are called as deletions, but even there's some points here and here, right? I mean, you might have thought these were duplications, deletions. It turns out, at least when I was running the HMM in this particular run, these didn't get called. And the reason that they don't get called, and this is an important point, is there are still lots of aberrations in read depth for particular exons, but the chance of calling any one particular exon is low unless you have neighboring exons. That is the point of that HMM. If you have 10 exons in a row that all have very low read depth or have very high read depth, then you're more likely to call a deletion or a duplication. So you still have exons. I think actually, we don't have to make a huge point of this, but where is that? Here. Yeah. I think if I actually show this here. So one of the important points here is that I didn't show this in the plot, just for simplicity here. One of the important points is if you actually look here, so I was talking here, these were the green were duplications, the red were the deletions, these were called deletions. So the gray are places that are called as copy number 2 or diploid. So if you look at the deletions, they don't actually look much different on average than the copy number 2. But the only reason, again, this is averaging over all targets. The reason that the deletions actually get called is because there are these runs they're contiguous, consecutive runs of low read depth right next to each other. If you just have low read depth at one target, that could just be dropout even after normalization, right? Okay. Sorry, so let's get back. Yeah, so anyway, yeah. Yes, I'll show some of those metrics soon that we had on at least real data where we had other basically validation data for those same samples. I'll get into that soon. When we actually talk about how CNV size, or at least in terms of number of targets, affects the sensitivity of the algorithm. So we'll get back to that soon. Just here, one of the points that I wanted to make is that so we used the hidden Markov model for each sample independently, as we mentioned, to call copy number variation for that particular sample. And again, here it's just plotted all the samples together. But one of the questions you might ask is, how sure am I of one particular sample? What are the quality metrics? What's the quality score for that copy number variant that I've detected? And even more so, how sure am I of the breakpoints? How sure am I of particular subregions of that CNV? And those are all questions that we've addressed using the hidden Markov model framework, and we've implemented that in XHMM. And what that actually produces are lots of different quality scores, and I'll detail some of those soon. The point is just that you can use XHMM to not only make the CNV calls, but you also get lots of quality scores that help you filter those and compare those between different samples. Just as a first test case on real data that we actually did at the time was we used trio data, which means we sequenced both mother, father, the mother, father, and a child. In this case, it happened to be a disease study, so schizophrenia trio, so meaning the child was affected with schizophrenia. And here we're using 90 trios. This is just an example where we actually find what looks like a de novo deletion in a particular gene, and that's actually been called previously and validated using Affymetrix array-based data. Fine, so that just tells us, okay, anecdotally, it seems like we were doing the right thing. We're starting to call deletions that have been called previously. Looking at a more global scale to get a statistical feeling of what's actually happening with the data. So what we do is, since in a disease study, we're pretty focused on looking for rare copy number variants. And to filter out any additional artifacts, we basically filter at a frequency of 5%. And that's not a hard threshold. The point is just, again, to get to the pretty rare events. So that's where we're focused, again, for disease. That seems to be where disease risk lies. So that's sort of the focus of XHMM. So what we actually see is, again, now this is pretty trivial, the plot on the left, but in conjunction with the plots that I'm going to show, you'll see why it's important. So what we see is that as we filter on increasing CNV quality, the number of CNV events decreases. Again, that's trivial. You filter at a higher level, you'll get fewer events. Moreover, pretty similarly, if you filter the CNVs at a higher quality threshold, you'll also get fewer de novo events. Again, these are a trio data set, so we have the advantage that we can start looking at transmission data from parent to child and see, does the child have events that aren't called in the parents, and those would be de novo mutations. So that we ask what happens as we have increasingly higher thresholds on the CNV quality that we require. So not only we have fewer events, but we also have fewer de novo events. Again, almost pretty trivial, but I think what really shows the power of the filtering and the quality scores is that as you filter at higher quality here, when we're looking at the trios and we look at transmission data from parent to child, and we'd expect, right, on average, just Mendelian transmission tells you that approximately 50% of events should be transmitted from that the parent has, should be actually transmitted to the child. So we see that if we filter at a high enough quality threshold, we actually get to that 50%. And it's pretty stable once you start filtering at higher and higher thresholds. So it basically tells us, if you filter the data, the calls, we have a pretty expansive call set. So not every CNV there is necessarily real, and not even after filtering. But what happens is that as you filter more and more using the quality scores that we provide, you get fewer CNVs called, which again makes sense, fewer de novo events. So that, again, we don't expect most events that a child has to be de novo. Most events should actually be inherited. But we actually also see the converse, that the events in the parents are mostly are transmitted at an expected 50% rate. So basically that's telling us overall the data seems to be telling us that XHMM is doing a pretty good job at calling rare copy number variants. So now moving on to a slightly larger set where we have more validation data. So at the time, again, we had approximately 1,000 whole exome sequences of a larger part of our Swedish schizophrenia case control study. Sequencing was done here. So we had looked at the XHMM call set on these 1,000 samples. And what we saw is that if you look at rare CNVs, there's approximately one coding deletion, one coding duplication per individual. So 2 rare CNVs per individual. How does that stack up in terms of do these CNVs validate? Are they real CNVs? So what we actually had for this same data set, we also had Affymetrix-based calls. So it's from a GWAS chip, so an array-based CNV calls for the same sample of 1,000, the same cohort of 1,000 samples. And we subsetted that down to those CNV calls that were done genome-wide to those that actually overlap some exome target. And looking at those calls, there were approximately 500 of those calls. The question was, how sensitive is the XHMM calling, the exome sequencing-based calling, to those genome-wide calls? So of those 544 calls, approximately 60 to 70% of those were actually sensitive to calling in our initial call set. And when we looked a little bit deeper into that data, what we actually saw, and this is sort of, I got messed up a little bit, okay. What we actually saw is that one of the important points is that as you increase or actually change the number of exome targets that the CNV overlaps, you'll have more power to detect the CNV. So this is actually looking at, as before, we're looking at the longer, greater than 100 KB, so that's 100,000 bases, CNVs from the Affymetrix data, or Affymetrix array data, and now we're asking how many exome targets do they overlap. So if it overlaps 1 target or more, which is the default, which is what we're looking at, there's 67% overlap. If you get to 3 or more targets, you already get to 80%. So if there's a CNV, a pretty long CNV that called by Affy, or reasonably long that we believe from the Affy data, and it overlaps the exome by 3 targets, meaning there's reasonable expectation that you should be able to call it, so 80% of those were actually able to call. And that's a pretty good number considering that even in array-based CNV call sets, there's pretty large divergence sometimes. So 80% was pretty good sensitivity. We were pretty happy with that. Obviously, if you have more exons that overlap that CNV, meaning you should have more power to call that. And that's what we actually see. If you go to 10 exons, if there's some CNV, it doesn't have to be even long, but it has to be long enough to overlap 10 exons. So it depends on the gene size, how big the CNV really is. So then 92% of those we're going to see. And there does seem to be some sort of like thresholding effect there, in that there are certain CNVs that we're just not able to call no matter what. Some of that has to do with the filtering that we do of certain exons, but there are certain regions that are probably harder to call. I think I'm not going to go into this in the interest of time. Let's sort of skip this. So yes, so I think one of the important points is, besides that, we're able to make the XHMM CNV calls themselves. We actually give a lot of different quality metrics, and they're useful for different types of scenarios, different data scenarios that you're actually using for, including the trios, and I'll explain about that in a second. So some of the metrics that we give include this exact delete, if you're talking about a deletion, for example. So it gives the EQ, which is sort of the exact quality. So it'll tell you what is the probability, or really a Fred-scaled probability. So it gives you a quality score telling you how sure are you that this exact event, if you have 10 targets deleted, it tells you how sure are you that these 10 targets are deleted in that sample as is. Another possibility that you might be interested in is, fine, I called a CNV that's 10 targets long. How sure am I that there's at least some deletion, at least one of those targets really is deleted in that sample? Sort of conversely, you might be interested in knowing, I found this in one sample, I want to know, does some other sample not have the event? And I can genotype that, basically. And I can ask, what is the probability of that other sample not having a deletion? Going back to the other case where you have a particular call in one sample, you might want to know, how sure am I of the left breakpoint? How sure am I of the right breakpoint? How sure am I that the sample is not diploid? So I don't know, maybe deletion, maybe duplication. How sure am I that they are diploid? Again, sort of asking, I found a CNV in one sample, I want to know, how sure am I that the other sample does or does not have the CNV? Just some examples. And again, those are Fred-scaled quality scores. Just as an example, I think, so this is on the bottom here, it says, if you want to call de novo CNV, so what would you do? How would you use these quality metrics in the right way? And I'll talk about that a little bit later, if we have some time. So the point is that what you would actually do, and this is the way we've implemented it, in the child, you want to call de novo CNV. So this is a CNV that you expect to, by definition, you want to see it in the child, but not in the parents. So you'll ask, in the child, is the SQ, the sum quality, is there some deletion in the child above some threshold? But you also want to require that in each of the parents, you don't see that deletion. So you'll require that the NQ, the no deletion, no duplication, whatever you're talking about, is high enough in each of the parents. And then you can be pretty sure that the data actually supports a de novo event, as opposed to sort of what you might think to do, at least in a more naive implementation. What you might do is just say, if there's a call in the child but not in the parent, I'll be happy and I'll call it a de novo deletion or de novo duplication. The problem with that is you end up with a really large number of false positive de novo events. And the way we've gotten around that, or sort of use the data more properly, is to actually use these quality metrics across all the samples. And just, this will relate to what I go through pretty quickly soon, but just that this all relates to the genotyping module of XHMM, where you can actually calculate all of these quality scores for every single event that you've discovered, but you'll calculate these quality scores in all samples that you have, even if the event was discovered or was not discovered in that sample. Just another example of where you might want to use the quality scores to sort of get at interesting biology, and we've actually done this, at least preliminarily. So if before we talked about deletions and duplications, you might have a non-disruptive event, and that was what I was showing in the cartoon originally, that in a non-gene disruptive event, you have the reference chromosome where you have the whole gene, and deletion, you're just missing that whole gene, and duplication, you have 2 copies of the gene. But you can imagine that in sort of what we call a gene disruptive event, in the reference, you have the whole gene, in a deletion, you deleted only part of the gene, in a duplication, you duplicated part of the gene. And those, we call them disruptive only because they might be more disruptive. You can imagine for disease, they might have a stronger impact if you leave around parts of a gene, think of gene fusion events, things like that. But in terms of actually detecting these, how to use XHMM to do that, you would actually want to, so for non-disruptive events, you would just ask, how sure am I that the particular event is real? Meaning, is there really a deletion in that region? so that you would just look at this SQ. Is there some deletion in that region? Is it above some threshold, that score? If you're asking about a disruptive event, not only do you want to know that there is a deletion in that region, but you want to, let's say, be sure about the left breakpoint if that's in the middle of a gene, or the right breakpoint if that's in the middle of a gene, and think of the functional consequences of that. We actually did that for this, at least this initial data set of 1,000 schizophrenia samples, and we actually detected there what seemed like a greater burden of disruptive events in cases versus controls, and we're still working to follow up on that in our larger set, which is now already at 5,000 samples. But the point is that using XHMM, you do have the power to try to look for these types of events and pretty sure look at disruptive events and not just having a CNV, but rather do I believe the breakpoints being at the left side or the right side, and do they interrupt a particular gene? So quickly, in I guess the 15 minutes or so that I have left, what I'm going to go through is how do I actually run XHMM. So let's say you have your own data set and you actually want to run XHMM. What do you do? All these slides are going to be online. It also matches sort of the tutorial that's available online, and also fine. Let's see. So what you actually need is the XHMM software and the GATK software. Those are the main two tools, and you also can use the PlinkSeq software for certain steps if that's useful. And again, this follows the tutorial. So you install XHMM. It's a C++ software package. So you download it and install it as follows. Basically, the input files, and that's the first step, is you need to define your exome capture interval list. And you use that in the sort of GATK standard of something interval list, and it has all sorts of listing of all the, let's say, 200,000 exons. The next sort of ingredient that you need to run XHMM are the BAM files, the analysis-ready BAM files that Bob referred to before. And that has the sequencing reads. And then you need some small file that has a few parameters for XHMM, and those can be tweaked, but sort of most people just use off-the-shelf parameters unless they have a very different setup of targeted sequencing or something like that. That's not similar to exome sequencing. So the first step is you're going to run GATK depth of coverage walker, and you can do that in parallel. In this example, and the one I have online in the tutorial, you run it in 3 different groups. Again, this is just the parallelization is just to speed things up. And literally what you do is you run the depth of coverage walker, you get out lots of different files. The main column that we're interested in is, in this case, again, this follows the tutorial online. So if you can't see anything here, you can always go back to these slides or to the tutorial online. And basically what you see is that the column that we're interested in is sample underscore mean coverage, which is the mean coverage for a particular sample for each of the targets, which is what you have in the rows here in this screenshot. So now that you've run the depth of coverage, you need to combine those mean coverage values into this matrix that we have here, into some real file that's a matrix. And for that, you run the XHMM command merge GATK depths, which literally takes the, yes, sorry. This one over here. Yep.
I will automate to the group 1 sources. So if I use this command, it will create a shell like room shell. They feel like you have so long command line.
Yeah. So there's actually another parameter here. It's not described on the slides here, that you can give it a file that lists all of the files. I'm pretty sure, I can't remember the name of the, yes, okay. But exactly, that is an issue. Exactly. This is just more, like when I actually run it, I usually run with a file that lists those things, or either way. But yes, exactly for that sort of point, to not have command lines that are overly long. Exactly. So basically, you run the merge GATK depths module. You end up with a file that actually represents this matrix numerically. One of the next steps, and this is one of the steps that I left out before, is we do some filtering. And the reason you do the filtering is because before you run PCA for the normalization, the principal component analysis, you want to get rid of any targets or samples that are very extreme and would sort of skew the PCA analysis. And so in this case, we're getting rid of targets that have either very high coverage, sort of outliers, or samples that have very high coverage or very low coverage. And of course, the parameters that we give on the XHMM website are sort of standard practice, but it depends, of course, on your exome sequencing setup. Depends what depth you're sequencing at. In this example, and the examples online, it's sort of assuming that you've sequenced at 100x on average or something like that. Again, now in terms of deciding which targets to filter out, so that you can decide that up front, and we do that using a mix of basically GATK and PlinkSeq, where you might decide to remove a relatively small number of targets that have very high or very low GC content. Just again, because we know that the capture isn't going to work so well there, or the read depths might be very skewed there. It's going to be hard to call the copy number variation based on read depth there. So we sort of remove those up front. Similarly, in terms of mapping, if there are targets that have very high fractions, at least a quarter of their bases being repeat masked, so if you just download the track from UCSC, or you look at certain reference genomes have that, and that's what we use PlinkSeq for in this case, then we actually just remove those certain targets. Again, these steps are optional, but they help with the data being cleaner and getting better results. And literally, so now we use the matrix command, which lets you do all sorts of different matrix operations in terms of removing rows, removing columns. So it's removing samples and removing targets. And in this step, you also remove targets and targets that are very short, or targets that are too long, samples that have high read depth or low read depth. Same thing for targets. And similarly, you can explicitly specify targets with extreme GC content or very low complexity, meaning high number of repeats. And that's where we remove that before the PCA step. In the next step, we actually run the PCA. So we split the filtered read depth matrix into its principal components, into the variance matrix, and to the sample loadings. And that's using the PCA command. You end up with these basically 3 files. Again, now how do we decide which principal components to actually remove? So before we talked about it in terms of like looking at the principal components, and some of these, or actually almost all these plots you can get from, there's accompanying R scripts that we have online that you can actually use to plot these results. And if you do look at them and you decide that you want to manually decide how many principal components to remove, you can do that also. But in XHMM, the default is to use something that basically removes relatively high proportion of the variance explained by the first K principal components, and it decides which K based on the percent variance explained. So now, once you decide how many principal components to actually remove, and that's actually here in this step, in the normalized step, now you have to actually apply that normalization. So basically what you're doing is you're reconstructing a read depth matrix again based on the principal components, but where you're setting certain principal components to have 0 variance, basically to not contribute anything there. And effectively that's what you're doing. You're remultiplying out these matrices to get this normalized read depth matrix. And that's what we're going to use to actually call the CNVs. Just as the next step, again, we want to filter out some outlier targets. Even after normalization, there are certain targets that just have very high variance between samples, and it's probably good, and that's why we actually remove those. Just because those, targets will probably be very variable between the samples, and that's probably not indicative of real copy number variation. Again, it's still some residual noise. And again, we do that using the matrix command. And while we're actually running that matrix command, another thing that we do is we tell it to center the data and calculate a z-score, right? So, and that's the z-score that I was actually plotting before, and literally that's why it's 0 normalized and also standard normalized by its standard deviation for that particular sample. And so we've done the filtering, we calculate the z-score, and that's actually the data that we're going to use as input to the HMM to actually call. One of the next step is actually almost a technical step, is once we've cut down the normalized read depth matrix in all sorts of different ways, right? We've removed samples and targets that were too long, had too high extreme GC too very extreme GC content. We remove samples that have high read depth or low read depth. We want to actually do that to the original read depth matrix just for convenience, so later on we can actually use that for plotting and annotation of the CNVs to know what was the mean read depth. So we do that in this step, again using the matrix command. We just basically remove certain samples or targets that we've removed previously from the normalized data. Now in the next step, we run the discover command, and that's actually what's calling the CNVs. It's running the HMM to call CNVs from the normalized depths, and this is just, again, the schematic of the HMM that we're using, and there are only 3 states, the deletion state or duplication state, and those are CNVs, or the diploid state. And in HMMs in general, right, there's a transition probability and an emission probability, and the transition probability tells you what are the chances of jumping from diploid to duplication, etc. And those are parameters that go into the HMM, and that's in the params.txt file. And there are certain defaults there, but you might need to change those. Just quickly running through what these different parameters are. So I mentioned this earlier, that one of the things we take into account is sort of an exome-wide CNV rate. It doesn't mean exactly the number of CNVs to expect based on, for a particular exome, but it's sort of, you can use this to sort of scale the number of CNVs. If for some reason you're running XHMM and you think that the number of CNVs you're getting are too high or too low, you can just lower that parameter in your file or raise your parameter depending on what you want, and you should get more or less CNVs depending on how you change that. Again, similarly, if you look at the next parameter, is just the mean number of targets in the CNV. Again, it doesn't mean this will really be the mean in your actual data, but again, think of it as a scaling parameter. The mean now, the default is 6, but again, if you raise that or lower that, you get shorter or longer CNVs in terms of number of targets. Again, I mentioned this before also. This number 70 just means 70 kb, and it sort of tells you what you expect to be it's sort of, there's an exponential decay between neighboring exons in terms of how strongly you believe that the 2 exons should have the same read depth, and that will tell you how often they should have a deletion CNV should continue or a duplication CNV should continue, or maybe they're just independent. If they're far enough away, at an extreme, if they're on 2 different chromosomes, then you don't really want the CNV at the end of chromosome 2 to affect the CNV at the beginning of chromosome 3. So that parameter sort of tells you how far you should go in terms of that. Then, right, if we're looking at z-scores, we have normalized z-scores. So the distribution of normalized read depths for the deletions. So right now we parameterize that as a normal distribution centered at -3 in z-score space with a standard deviation of 1, again, because we did z-score normalize. For the diploid state, we characterize that with a normal distribution centered at 0. And for the duplications, we characterize that as a normal distribution centered at 3. Again, these are parameters you can change in the params.txt file. The default is online, so you can use that. And it seems to work pretty well in different scenarios, but this is something that you can change pretty easily. It's a pretty small file. One of the nice things I put into XHMM, I thought, was when you actually run, it actually prints out this thing on the bottom that tells you what the parameters you're using actually translate into. And it gives you that output right away. So if you change these things, you can actually know pretty quickly what they mean. And sort of the last step in the XHMM pipeline is what we talked about earlier, the genotyping step, which actually outputs a VCF file with call genotypes. So these call genotypes are a little bit different than what Bob was talking about, because here we're only calling really 3 possible states for each sample, either diploid, deletion, or duplication. So we actually call them, here it just says haploid, and it's in quotes because they're not necessarily really haploid. The point is every sample will only have 1 genotype, either diploid, deletion, duplication, or one of the important points here also, they can have a no call, so they'll have a dot in VCF format, which basically means based on the read depth data, we're not actually sure what they have for that particular region. So they'll have diploid, deletion, duplication, and what the genotyping step actually does is it takes the CNVs, every single CNV called in the discover step, and genotypes those and asks what the hard call is, diploid, deletion, or duplication for each of the samples. But importantly, not only does it give you the hard call, it also gives you all those metrics that we discussed earlier for all of the samples. So if you want to ask, is this a rare event in one particular sample, am I sure that none of the other samples have it? For example, when we're talking about the trios, if you're looking at the child, then you can also get, as sort of a side effect, you also get in that same VCF, you'll get do the parents have that event? Do other children, affected children, unaffected children? If you have a large pedigree, that might be useful also. So you get basically the genotypes and the metrics for all the samples. As I mentioned also, one of the things that come with the XHMM software are these plotting scripts in R. And this is just an example figure that you can actually make using those scripts. Again, these are online and the part of the tutorial, and even in the tutorial example, you can actually run it and you should be able to get one of the CNVs, or the two CNVs that are called in that the tiny toy example plotted. And that's exactly this example data set. There is really a toy data set from 1,000 genomes, whole exome sequences, just for a very small data set, 300 targets on chromosome 22, but it already gives you a feeling of how relatively easy I think it is to run XHMM if you follow all the steps literally as they are online. In terms of what's next, so okay, you called CNVs, how do you actually use the CNVs called by XHMM for analysis if you're doing disease analysis or disease study? So one of the things we have written and implemented as part of the PlinkSeq software, which we hope to be documenting a little bit better soon, but there's the CNV de novo module, which exactly uses the VCF in the way I've described to look for de novo CNVs, transmitted CNVs. That was basically how I did all of that analysis. We wrote this tool in PlinkSeq that reads in the VCF from XHMM and does all those types of analyses. There's also a sort of helper script that comes with XHMM that literally converts the CNV calls from the discover step to Plink's CNV format. And since Plink has a lot of functionality for CNV analysis, including case control association, so that's that's one path to go forward. So you take the XCNV file from XHMM, convert it to Plink's format, and then you use Plink to actually just do the CNV association if that's what you're trying to do. One of the things we're working on, I mentioned before about the gene disruptions and using the VCF from XHMM to look at the left breakpoint and the right breakpoint. Of course, if you want to look for gene disruptions, you have to overlay that with locations of genes. And the way we're doing that, again, is in PlinkSeq. And that's software that we're working on and should be released as a part of PlinkSeq pretty soon. In terms of actually downloading the XHMM source code, so it's in the Bitbucket repository. And if you have questions in general, you can basically email the XHMM-users forum, which is a Google Groups here at the BroadE, but it's also, you can also ask questions on the GATK forum. There's also an XHMM page there. And one of the important points was also for parallelization. I'm not going to go into details right now, but there is one Q script that lets you run the full XHMM pipeline basically as described on the tutorial using this Q script, and it's part of actually, this slide is a little bit out of date, it's actually part of the GATK release for the past couple of months. So if you're using GATK, the full release, then you can actually get the XHMM Q script and run XHMM, the full pipeline. Basically the input is BAMs and an exome target list, and the output would ultimately be the CNV calls and the VCF that gives you the genotypes for all samples. Yeah, so I think we're just over time now, so let me finish up. Just overall, I guess our conclusion was from using the exome sequencing data, it wasn't clear going into this sort of project that you could use exome sequencing in a realistic way, sort of, to get CNV calls, but we showed that we were able to, and other groups have also shown that over the past 2 years. XHMM is one of the tools. I think it has a lot of flexibility, and it provides a lot of metrics, so I think that's a useful thing. And some of those metrics let you look at new biology, including gene disruptions, de novo events, in a sort of robust way. That's just about our publication, and some acknowledgements for lots of people here at the BroadE, at Mount Sinai, that have worked with, and our collaborators, a lot of them in the UK and North Carolina that we've worked with on different data sets and helped us really develop XHMM, especially in a disease context, for looking for rare CNVs. So I think that's it. We think we're over time, but if anyone has questions, you can ask now, or we can just end and you can ask me in person too. Thank you. Yes?
For
Yes.
Because I don't know the background knowledge to set up particular number, how to set up the file.
Yeah, so you're talking about the params.txt file. So
Based on random sample, or based on average or based on any.
which number are you talking about, or?
Like
All of them. Yeah, I mean, part of what we did, I mean, I didn't go into that here. What we did is to get to these parameters on different data sets, we basically, at the time, we were using actually trios from some of the autism sequencing. And we actually sort of did a grid search for lots of different values of these parameters simultaneously and seeing where did we get the best results in terms of some of the trio, again, using the trio analysis in terms of transmission, not having too many de novos and having 50% transmission. And this is what we came to. Again, we still do the post quality score filtering, which I think is an important point, is that if you're running pretty standard exome sequencing here at the BroadE or in other places, these parameters usually just work. And the more important point is to filter downstream using the quality scores that come out. So again, for any particular project, it might be important to change these parameters, but I haven't found that, and we've run it on a bunch of different data sets in different ways. Usually what's more important is choosing a good threshold to filter on later for downstream analyses. Because Exactly. I think that's the way most people treat it, and it's, again, I think like a lot of programs, including GenomeStrip, but also all genomics programs have lots of parameters, and as the developer of a program like this, you need to give users the option to change those parameters, but I know from personal experience, but also from experience here, most people aren't going to change those parameters very often, unless they have some crazy results that are coming out of the software, right? So.
And another question is like the software package has to be based on coverage and read the particular region. Why do you compare the base quality? If you have like a very high margin quality read coverage state, it's that command you read this case exactly the A and the base quality. So why base quality is
Yeah, okay, so we've thought about that, and there's different combinations, and it also has to do a little bit with the way the base quality and mapping quality thresholds are implemented in GATK, but another point is also that part of the mapping quality is for the read as a whole, but if you're really not sure of the base, how certain can you really be of the mapping of that base to the genome also? You know what I mean? You're right, if there's 50 bases here that map really well and 50 bases here that map really well, it doesn't really matter what that one base is in terms of that there is some base that covers that part of the genome, the exome, really. But on the other hand, right, I mean, it could be if that base doesn't match the genome. If let's say it's a base quality 0 or 2 or whatever denotes. Yeah, but not, I mean, maybe, but I mean, you could have some other, if that base, if all the other part of the read maps perfectly, but that base, you're not even sure what that base is, can't know for 100%, right, if it actually maps to the genome perfectly or not. And there could be some other underlying event. Yes, Bob? Here, maybe Bob can help me out here, yeah.
I was actually just going to ask the question a different way. So do you know empirically how important the base quality is on top of mapping quality?
Yeah, I'm not sure. We did some of these trials a while back where it seemed actually that even the mapping quality filter, which is sort of like the default that we recommend, didn't seem to make a huge difference. Again, we do some other filtering, right, in terms of GC, very extreme GC targets with very extreme GC content or high repeat mass sequence content. So those could be sort of covering for the fact that mapping quality might not matter later on. Plus, we do a lot of post-calling filtering, and that could be also where some of these missed mapping regions cause problems, but then we get rid of it later on. But it didn't seem to make a huge difference when we filtered the mapping quality or not. Like we were looking at the transmission, and it didn't seem to make a huge difference. We could set the mapping quality threshold to 0 as opposed to 17, I think, which is the default now, which is sort of like the GATK calling threshold, and it didn't seem to make a huge difference. So that's why I'm not so troubled about that concept, but that's also why I think that you might need to play with that for your data, I guess. Again, it's another, there are lots of thresholds here for the data, but you're right, I don't know 100% why, or I can't tell you 100% for sure that you don't need to, that you should filter that and not the other. Again, it depends, sort of, yeah.
Maybe it's more important that it's unbiased.
Yes, exactly. Yeah, exactly. Yes?
So I will try to use the first 20 principal components and see if they correlate with genotyping done on the exome, but I'm talking about SNP genotyping, not the CNV genotyping.
So you mean common? Are you talking about frequency based, or? I mean, because those strongest principal components, the way they look, if you actually plot them, often look like the ones of the. If we go back a lot here, if we go back to the, they often look like these ones, meaning that they're these very strong components that are scattered throughout the genome. So it would be hard for it to be, because of the LD structure on the one hand, it would hard to be like due to.
I was thinking more as a quality control for the genotyping. Maybe there's some genotyping that is artifactual. Maybe it has to do with some of these.
I see what you're saying. In other words, if the read depth, I see. So you're saying there could be regions where the genotypes that come out of the exome sequencing might be due to some strong principal components in certain parts of the genome. The thing is, it's possible, but again, if you get a principal component like this, how would you interpret that? Like, would that tell you that the whole, like, this would say like half the genome or half the exome is possibly poorly genotyped or something? You know what I'm trying to say?
I think maybe there is an exome that is not very well covered, and maybe
yes. That could be, you're right. So, some of these principal components that we're doing for the data could inform back on the QC. I think the QC that we've done, at least on these types of data sets for the SNPs, is probably even more stringent than the principal components would tell you about those samples. But there are points where we did go back and look which are the samples that had very low read depth, and we did filter them out. But again, that's almost something that's trivial to check otherwise. But it did correlate with other QC metrics in the SNPs. High, let's say for males, looking at high chromosome X heterozygosity or things like that. There are sorts of artifacts that we did see that correlated with read depth based metrics, yes. So the answer is we haven't done a complete analysis comparing the two, but there are definitely anecdotes where we did see some overlap between the two worlds. One of the things that we actually want to still implement, and we think just in terms of mapping, right? If you have a SNP, or if you have multiple SNPs in an exon, which might not be that common, but even having one SNP in an exon, that the mappability of that, the number of reads that map, just because there are reference biases along the way, but also bioinformatically there are reference biases, that will decrease, it could slightly decrease the number of reads that map to that exon. And when we're using read depth, right, that can affect some of the noise that we see, some of the decreased read depth that we see throughout the exome that looks like noise might also be just due to having single nucleotide variants in that. But that doesn't affect you necessarily from having a deletion on the opposite strand or even on that strand, right? Or a duplication, let's say, right, on the other chromosome. So taking into account the SNP calls when we're trying to model the read depth is one of the things that we've been planning on doing for quite a while, but we actually haven't gotten around to it yet. But that's just in terms of intersecting the SNPs and the read depth information. Yeah. But you're right, it's a good point. Right now, we're XHMM is now very focused on read depth, but sort of intersecting that with the SNP data would be a good idea still, yes. Okay, thank you.